Bismillahirrahmanirrahim. This is Muhammad Huzaifa from the Department of MLT, Cohort University of Science and Technology. Today I will present normal flora. In the content we will discuss about definition of normal flora, species of normal flora, advantages and disadvantages, symbiotic relationship, types of normal flora, resident flora, transient flora, tissue specificity of normal flora, tissue tropism, specific adherence, biofilm formation, normal flora and human body scan, eye, oral and upper respiratory tract, gastrointestinal tract and rectum, disease caused by microbiota, fungi, viruses, conclusion and reference. Inshallah, we will discuss about all these things. Definition: the term normal microbial flora denotes the population of microorganisms that inhabit the skin and mucous membrane of healthy normal person from shortly after birth until death. Their flourishing depend on physiologic factor of temperature, moisture, and the presence of certain nutrients. Species of normal flora: first one bacteria and second fungi, the third one viruses. Advantages of normal flora: first one, normal flora provide a first line of defense against microbial pathogens through bacterial interference. Interference may involve competition for receptors or binding sites on host cell, competition for nutrients. Assist and digestion: they produce vitamin B and vitamin K and intestine. Play a role in toxin degradation. The oral flora contribute to immunity by inducing low level of circulating and secretory antibodies that may cross react with pathogens. The microbiota affect carcinogenesis in three broad ways: altering the balance of tumor cell proliferation and death, regulating immune system function, influencing metabolism of host produced factors, food and pharmaceuticals, such as lactobacillus and bifidobacteria are known to prevent tumor formation. The disadvantages of normal flora: they can cause disease in the following. When individuals become immunocompromised, when they change their usual anatomic location. Symbiotic relationship: mutualistic, both organisms benefit, mutually beneficial. For example, Escherichia coli, E. coli synthesizes vitamin K and B complex vitamin. In return, we provide a warm, moist, nutrient-rich environment for E. coli. Commensalistics. The second word is commensalistic. One organism benefit, the other is neither helped nor harmed. Opportunistic. Under normal condition, microbe does not cause disease, but if condition become conducive, it can cause disease. E. coli normally in our digestive tract where it cause no problem, but if it gets on the urinary tract, it can become pathogenic. Staphylococcus aureus, commonly found in the upper respiratory tract, but if it get into a wound or burn, it can become pathogenic. Types of normal flora: resident normal flora, transient normal flora. And resident normal flora, microbes that are always present, and transient normal flora, microbes that live in or on your body for a period of time, or for example, hour, days, or week, then move on or die off. Transit microbiota remain in the body for only hour to months before disappearing. Found in the same region is resident microbiota cannot persist in the body because competition from other microorganisms, elimination by the body's defensive cell, chemical or physical change in the body. If the resident microbiota is disturbed, transient microorganisms may colonize, proliferate, and produce disease. Tissue specificity of normal flora. Most members of normal bacterial flora prefer to colonize certain tissue and not other. This tissue specificity is usually due to property of both the host and the bacteria. Usually specific bacteria colonize specific tissues by one or another of these mechanisms. The number first, tissue tropism. The number second, specific adherence. The number third, biofilm and biofilm formation. Tissue tropism as the bacterial preference for certain tissue for growth. One explanation for tissue tropism is that the host provides essential nutrient and growth factor for the bacterium, in addition to suitable oxygen, pH, and temperature for growth. Lactobacillus acidophilus, and formerly known as Doderlein bacillus, colonize the vagina because glycogen is produced where provide the bacteria with a source of sugar that they ferment to lactic acid. Specific adherence. Most bacteria can colonize in specific tissue because they can adhere to the tissue in a specific manner that involve chemical interaction between bacterial surface component and host cell molecular receptor. The bacterial component are molecular part of their capsules, fimbriae, or cell wall. The receptor are human or usually glycoprotein molecule located on the host cell. Example of bacteria specific adherence to host cell or tissue. For example, bacterium. The number one is bacterium. The number second is bacterial adherence attachment size. Streptococcus pyogenes is adherence to cell bound protein, so we call M protein. Pharyngeal epithelium is the attachment site. Streptococcus mutans cell bound protein glycosylic transferase through glycosylic transferase enzyme attach with the pellicle of tooth, and there is some other example of the bacterial specific adherence to the host cells or tissues. Biofilm information. Biofilm formation. Some of the indigenous bacteria are able to construct biofilm on a tissue, or they are able to colonize a biofilm belt by another bacterial species. Biofilm usually occur when one bacterial species attach specifically or non-specifically to surface and then secrete carbohydrate slime that embeds the bacteria and attract other microbes to the biofilm for for the protection or nutritional advantages. Sterile tissue. In a healthy human, the internal tissues such as blood, brain, muscle, cerebrospinal fluid. Cerebrospinal fluid are normally free of microorganisms. Normal flora in human. Number one is skin, eye, oral and upper respiratory tract, urogenital tract, gastrointestinal tract, and rectum. Skin Staphylococcus epidermidis, Staphylococcus aureus, Micrococcus species, Corynebacterium species, Peptostreptococcus species, small number of other organisms, Candida species, Pseudomonas, etc. Oral and upper respiratory tract, diploid, non-pathogenic Neisseria species, hemolytic streptococci, Staphylococcus epidermidis, and non-hemolytic streptococci. Anaerobic, too many species to list. Larger amount of the following when accompanied by organism listed above: yeast, Haemophilus species, pneumococci, gram-negative rods, Neisseria, gastrointestinal tract and rectum. Various enterobacterioids except Salmonella, Shigella, Yersinia, glucose non-fermenting gram-negative rods, enterococci, alpha hemolytic and non-hemolytic streptococci, diploid Staphylococcus aureus and small number, yeast and small number, anaerobic and large number, genitalia. Corynebacterium species, Lactobacillus species, alpha hemolytic and non-hemolytic streptococci, non-pathogenic. Neisseria species, enterococci, enterobacteriaceae and other gram-negative rod, Staphylococcus epidermidis, Candida and other yeast anaerobes, too many to list. Conjunctival flora. The conjunctival orbits few or no organism. Haemophilus and Staphylococcus are among the genera most often detected. Disease caused by microbiota or normal flora. For example, Streptococci of the Viridian group are the most common resident organism of the upper respiratory tract. A large number of them are introduced into the bloodstream. They may settle on the deformed or prosthetic heart valves and produce infective endocarditis. Bacteroid species. Bacteroid species are the most common resident bacteria of large intestine, quite harmless in that location. However, if introduced into the peritoneal cavity or into pelvic tissues along with other bacteria result of trauma, they cause suppuration and bacteremia. Some genera of bacteria such as bacteroides and Clostridium have been associated with an increase in tumor growth rate. Fungi. Fungi is a particular yeast or present in the human gut. The the best study of these are candidate species due to their ability to become pathogenic and immunocompromised, and even in healthy host yeast also present on the skin, such as Malassezia species, where they consume oil secreted from the sebaceous gland. Viruses. Viruses, especially bacteriophages, colonize various body sites. These colonized sites include the skin, gut, lung, and other oral cavity. Virus communities have been associated with some disease and do not simply reflect the bacterial communities. The conclusion of the normal flora. Normal flora are microbiota colonized in human body since birth until death. Some species are benefit to humans such as defense against pathogen, produce vitamin K and B such as known as mutualistic. In certain situations may become pathogen, especially when change normal region or um, or immunosuppression opportunistic. Microbiota consists of bacteria, fungi, and viruses, and common sites where colonize are skin, mouth, respiratory, gastrointestinal tract, eye, and genital. Some region sterile, no microbiota, which are blood, brain, muscle, and C safe. Tissue specificity is usually due to property of both the host and the bacteria. So this is the end of the normal flora. अगर ये वीडियो आपको अच्छी लगी और लेक्चर अच्छी लगी तो आप लोगों से गुजारिश है कि आप लोग इस चैनल को सब्सक्राइब कर दें और दोस्तों के साथ शेयर कर दें। शुक्रिया। अल्लाह हाफिज़।